3, 4, and 5. Out of that, almost like half of it is affected. And then again, this particular one has got 1, 2, and 3. Again, half are affected. Here, half are affected. And therefore, this is purely dominant, autosomal dominant trait. Whereas, look at here. This is an incomplete dominant or semi-dominant, and therefore this individual also got it. But look at that individual is very severe, and he essentially passed away. Okay? So that is what is it called, incomplete dominance. But technically, no dominant disorder is purely dominant disorder. Okay? Even if you take very carefully, molecularly, if you go back, let us say a dominant disorder, you have an enzyme which is deficient. According to the inheritance, you have half, right? Half should be affected. So you go back to these half individuals in a family and then look at actual levels of this enzyme. If you compare the levels, let us say it should be essentially 10% or something, the null frequency. But you will not get that 10% in both of them. One of them may have 10% and one of them may have 15%. So if you look at a molecular way, no disorder is purely dominant. You can still classify them as incomplete dominance, okay? But what we are talking is the gross phenotype or gross morphological phenotypes or gross disease expressivity. If you look at those things, only then you can classify them purely as dominant. Okay? So even Huntington's disease, which is claimed to be a purely dominant disorder, sometimes you will have differences and you can tell that. Achondroplasia is an incomplete dominant disorder. Skeletal disorder, like, this is a skeletal disorder with a short-lived dwarfism, etc. Okay? So this is an achondroplasia. This is an autosomal disorder, and this occurred with a new mutation. Familial hypercholesterolemia, just now we talked about, that is an incomplete dominance. Okay? So look at that. This is tremendous nodules. That is real cholesterol. All sorts of nodules are there. These are hypercholesteremic problems. In general, when you go to a little bit higher levels of cholesterol, you don't get those. But this is heavy dose of cholesterol. This slide actually coming from UT Southwest. Now, if there is a new mutation, you should be able to distinguish whether it is inherited from heterozygous or from a new mutation. A new mutation can affect a fitness. So some could be genetic lethal, they don't reproduce at all. Most of the new mutations are genetic lethals. Generally, they're all genetic lethals. That is, reproductive fitness is 0. That's what is genetic lethal. Okay. Now we are going to talk about a different type, that is sex-limited phenotype in autosomal dominant disease. So there is a disorder called male-limited precocious puberty. But that happens only, secondary sex characteristics only in boys undergo adolescent age growth spur, almost like 4 years old. Right? So this is because of the receptor for luteinizing hormone is problematic there, and therefore signaling mechanisms can go haywire. They have normal fertility, multi-genetic pedigrees are well known, but this particular disorder is difficult to distinguish from X-linked disorder. Because of that, you have to do a mapping of the gene. This is a male-limited precocious puberty. It's only a 4.5 years old boy. Look at him. The muscles are essentially like an adult, and he has got puberty at that age, and this is a tremendously bad situation for his cousin. So if you look at the pedigree pattern of this male disorder, look at there, that is a male. It's just a male being affected. Only males are being affected. So, but carrier females are unaffected totally, okay? Therefore, it is definitely autosomal inheritance, not X-linked, because the trait is actually transmitted not by a carrier female. Carrier female is unaffected. So it cannot be Y-linked. So that's all we can tell about that. So now we're going to talk about X-linked inheritance. Males are hemizygous. Right? We talked about the term hemizygous. And females, only when there is a homozygous mutation, you lead to hemophilia. Therefore, female hemophiliacs are very rare. And we talked about how a hemophilic female can occur because of X inactivation problem as well. Yeah.
The prior diagram, how did you have, can you go back one slide? Why was there a proband in there?
This is the proband.
Why would it be higher up there?
No, after this fellow came and got diagnosed, then only this pedigree came out. You see, the proband,
okay.
you go back and analyze all the individuals. Okay? I just showed the pattern of inheritance there.
So
the
the
proband is the guy,
identified
first one who goes to the disease and who goes to the clinic and identifies the disorder, that is the proband, right? So that's why the arrow is there. Okay? So, the manifesting heterozygote, okay, but they can have, obviously you can detect it. You know, if you want to really go back and do molecular work, you can detect heterozygous. Okay, like for example, in a muscular dystrophy. Look at here, a normal female is having this muscular dystrophin. This is a dystrophin protein. They did immunohistochemistry on muscle cells. Okay. So when you do the immunohistochemistry here, you are obviously going to have all the cells lighting up with that dystrophin. Whereas a person with a disorder, okay, will have no staining at all for the dystrophin protein. Whereas this is a carrier. The carrier, first of all, it is going to be some cells. Okay, obviously, a carrier female will have some cells which are but low levels, half the levels of that. But on the other hand, if there is a problem with X inactivation situation, you will even have, because there is obviously X inactivated, a problem is there, you will have only half cells lighting up and half cells don't light up, and so on. But if there is a mutation along with the X inactivation issue, you're going to have a similar kind of situation like that. Okay? Recessive and dominant inheritance of X-linked disorders. So you have both recessive and dominant phenotypes. If the carrier expresses the phenotype, it is called dominant, X-linked dominant. If the carrier expresses the phenotype. Otherwise, it is considered recessive. Okay? This is an exceptional case where if X inactivation is rather skewed, you will have difficulty in identifying whether it is dominant or recessive. I think this slide is to let you know how confusing the nomenclature in genetics is. Therefore, I would not, in fact, what would you recommend because of this confusion? Look at here, 40% of all X-linked disorders are considered recessive. They said that first. Show no penetrance. And now they're telling 30% show penetrance and they're called dominant. Other 30% show some penetrance and in some not. Therefore, we cannot say whether it's dominant or recessive. So that is why there is a tremendous confusion in X-linked disorders. So what I would recommend is just get rid of the nomenclature. That's what others have recommended. So this nomenclature, just forget it. Whatever we talked about so far, the previous slides, use that. You don't go into too much details of that and get confused. Examples of recessive, classical hemophilia, which occurred in the Queen Victoria's family. Okay. So all daughters of hemophilia male are obligate carriers. Okay. And therefore all sons in the Queen Victoria's family got that, and that is how we knew this X-linked inheritance a long, long time ago. That's a classical disorder that you should remember. Affected male versus normal female, if you just cross them, all daughter carriers, all sons are affected. Normal male and a carrier female, half daughters are affected and half sons are affected. So this is a pedigree demonstrating the X-linked inheritance. The carrier is put as a dot inside. So a male obviously transmitted his X chromosome to that, and therefore she becomes a carrier. And in this case, obviously the carrier is there, and you are going to get one allele from her, therefore the male gets suffering. Therefore, if you look at the pedigree, if you have these kind of a closed boxes, only males, but females are unaffected. So you won't see it, right? You won't. Now that pedigree, you know that there's a carrier, but you have to find whether she is a carrier or not based on this pedigree. So male, male, and male, you think it is definitely an excellent recessive phenotype. But in the last few slides, we're also going to show you that there is a disorder which will tell male, male, male. It looks as if X-linked recessive, but it is a dominant disorder. Okay? So those things you have to be careful about. So what happens if there is a consanguineous marriage within this X-linked recessive phenotype? So you got a male to male, but then you cross it with a carrier female because it is a consanguineous marriage. It's a cousin marriage. And therefore you are going to get even a female hemophilia. Okay? So if a question is asked how a female can be generated as a hemophilia, you should be going with this answer, or you could do X inactivation answer. Either one is correct. Okay? Okay. There are various types. You can have a tremendous number of disorders of X-linked, Duchenne muscular dystrophy, hemophilia, Viscott-Hauser syndrome, X-linked immunodeficiency, color blindness, you can name it. There are a variety of disorders present on this X chromosome. Again, depending upon the disorder, the penetrance can vary. For example, there is one classical example. In this case, there is a disorder where the lysosomes are the ones which degrade the mucopolysaccharides, produced by the connective tissue. But there is an enzyme called iodouranate sulfatase. If there is a deficiency of that enzyme, you are going to cause the mucopolysaccharidosis, because mucopolysaccharides will not be degraded because there is a deficiency of the enzyme. And therefore, this is also called a lysosomal storage disease because these mucopolysaccharides will accumulate, and that is called mucopolysaccharidosis. But how does this enzyme act? This enzyme, what it does is, this enzyme can be taken up by the neighboring cells. You see, the cell produces it, but next neighboring cell can take it up. So even if the cell carries a mutation, let us say in a female, there are two X chromosomes. Obviously, some are inactive and some are active cells. But the active cells will produce this enzyme, but the inactive cells will take it up. Therefore, obviously, the mucopolysaccharidosis will not be there because even though the enzyme is deficient in some cells, you still can have those cells functioning just normally because of these neighboring cells will take up. Neighboring cells which are defective, they will take it up and therefore they will not show the disease. But in a situation like this, you can also have variable penetrance. So that is called Hunter syndrome. These enzymes are, if the cell having normal allele secretes the enzyme, the cell carrying mutant allele takes it up, then it is going to complement and correct it. Therefore, in these kind of cases, if the mutation also exists, the penetrance and the expressivity is varied because it depends on how these cells are located, right? If it is extremely closer to the cell which is producing, it's going to take it up. But if it's going to be interior, they're not going to take it up. And thus, the disease is going to be varied in expressivity and penetrance. Okay, same situation. X inactivation can occur for many of the disorders, like Fragile X syndrome, similar situation. Okay? There are only a few X-linked dominant disorders. That is X-linked hypophosphaturickets. This is the ability of the kidney tubule to reabsorb filtered phosphate is totally impaired. That is because of a gene called endopeptidase gene, which is actually controlling the hormones. And because of those hormones are not properly processed, you're going to have these kidney tubules being defective in taking the phosphate back in the system. So, in these X-linked dominant disorders, there is always male lethality. It occurs before birth, so daughters only are affected. Okay? So look at here. This is an X-linked dominant inheritance. The male is having a disease, but daughters got it, And again, this daughter is going to